This procedure aims to identify and measure prostaglandins and other eicosanoids in extracts of C. elegans. First, grow the worms in large-scale cultures. Then, using a liquid-liquid extraction technique, extract the hydrophilic lipids from the worm tissue. Inject the lipid extracts into an HPLC column coupled to a mass spectrometer. Then analyze the data from the liquid chromatography tandem mass spectrometry. Ultimately, this LC-MS-MS approach to purified worm extracts can identify new metabolites and also measure known metabolites, such as the F-series prostaglandins.
So the main advantage of this technique over existing methods, such as enzyme immunoassays, is that it is highly sensitive and specific, while at the same time comprehensive and flexible. Demonstrating this technique will be Hoang, a graduate student from my laboratory.
For supplemental feeding of the worms, pellet an Na22 bacterial culture by centrifugation. Discard the supernatant and resuspend the bacterial pellet in 100 milliliters of M9 buffer. Store the concentrated bacterial solution at 4 degrees Celsius. Start the worm cultures on 5 extra-large NGM plates. Continue to propagate the worms for 3 to 4 generations until the plates are full of gravid adults. To prevent starvation, add approximately 1 milliliter of the concentrated bacteria to the worm plates as needed. Air dry completely before placing the lid. Next, wash the gravid hermaphrodites off the plates with M9 buffer. Harvest the worms in a 50 milliliter conical polypropylene tube, then allow the worms to settle to the bottom. Discard the supernatant containing bacteria